yeah, this is incredible. I've definitely been to a lot of conferences and never anything like this. So just, I want to talk a little bit about what Illumina is doing, and first I want to, kind of give a brief overview, which many of you are going to be very familiar with, and this is mostly going to be a review for you, but not surprising to anybody in this room, but cancer is an incredible burden on the global population. Depending on what source you cite, if you're a woman, you have between 20% and 33% chance of getting cancer at some point in your life. If you're a man, you have up to a 50% chance of getting cancer. I mean, that's astronomical. I mean, that's staggering. Just to think about that. And we'll all have the chance to be impacted, by cancer at some point in our lives. So. So thankfully, advocacy groups aren't just the ones who are recognizing this or individual patients. Fortunately, folks with loud voices, people and politicians like Joe Biden are now putting effort and money into this. So I'm sure many of you have heard the Moonshot program, which is targeting or aims to infuse $1 billion of new research money into cancer research up through 2020. So I think this is an amazing step in the right direction. We're finally getting the attention of people that can really move a lot of this stuff forward, if for nothing else financially speaking. So again, not surprising, cancer biology is very complex. It is kind of a puzzle, but it's important to note that we've made a lot of progress too. It's a tough problem to solve. But we're working, every day we're getting better, we're understanding it better. And I just want to take a few minutes and kind of go through some of these categories. So all these are categories of active research, areas that we want to make progress on or in, and we have. And some of these have already been talked about today. So for instance, identification of driver mutations. We've heard that in some of the talks. What are driver mutations? These are mutations that actually cause the cancer. They're simply called passenger mutations, which don't lead to cancer. And it's very tough, even though it might sound like an easy concept, it's very difficult to determine the difference between what a passenger mutation is and a driver mutation, because you look at a sample and it's mutated, but you don't know if that mutation is the one that's causing it or if it's just correlative. Moving down, and this is the microbiome. So I know this is kind of an area of interest, active research and interest in, PMP. Obviously, we want to understand, and not just PMP, but this is actually taking center stage in a bigger way at places like Washington, where I come from. We want to understand how the microbiome is affecting health in general, including obviously cancer. So obviously a hot topic and area of research. Moving up to the top. The pathway analysis, as a few of the other speakers have already mentioned, there's kind of a long path between a mutation in your genome and all the way to the cancer itself. We want to know at what point to treat, what part of that to target, and how do we inform pharmaceutical companies, to start to create targeted drugs. Moving down to the tumor microenvironment. So this makes, this is one of the factors that make each tumor unique. We want to know what is surrounding, what surrounds that tumor, what's inside of it, what inflammatory cells are inside of it. We want to know that because that's going to help us understand it better, the prognosis better, how to treat it better. So anyway, there's a lot of factors that go in, just the environment, absence of genetic information, just the environment. And then, as also has been touched on, tumor subtype. Cancer is not cancer is not cancer. Your tumor is different from somebody else's tumor. They're very unique. It's almost like individual snowflakes. And what we're trying to identify is, okay, there are a lot of different types of mutations. Just like the speaker said, they looked at approximately a dozen patients and 2,173 mutations. That's incredible, right? But you want to find the common thread, underlying all that. You want to know the 10 mutations or so that are actually driving that. Moving on up, the role of germline variants. So, Sherry had talked about some this germline stuff. Obviously, we want to understand if there are any predisposing factors that you're born with that predispose you to developing cancer. We want to understand that at a very deep level. Gene regulation, so it's not just about mutations, it's also about how genes are actually turned on and off. It's kind of an interesting concept to think about. You know, it's not just how a gene is changed, you know, throughout your lifetime, but it's also how does it get expressed. You know, what are the proteins that are eventually, you know, made from that. And then the top, understanding drug resistance. Obviously, we want to understand, you know, if you're treated, you know, as a first round and you become resistant to a drug, we want to understand why, you know, that resistance is occurring and how to better counteract that. And then finally, of course, you know, ultimately, the ultimate goal is understanding the response to therapy. You know, that's the goal. You know, improving patient outcomes always. So that was a lot of talking. Oops, let me back up. So I would love to be able to talk about all those points, all those categories, but just not enough time. But you know, as I mentioned, you know, the first category, identification of driver mutations. It's really great that people are recognizing, you know, the importance of understanding driver mutations, and there are a lot of efforts that are underway currently to, you know, to better understand or better classify and identify what a driver mutation is. And it takes a lot of effort, it takes a lot of work, it takes a lot of money. So just for, you know, for instance, you know, in this particular effort, they've collected 3,000 tumor samples just to better understand 12 types of cancers. You know, and these individual subtypes. But this next effort actually is looking to collect 100,000 tumors, you know, to better understand these subclassifications of tumors and truly understand what's driving it, you know, what is really causing cancer. So obviously, there's a lot of work ahead of us, but we know, you know, we're finally on the right track in a lot of ways. You know, we're finally going down the right, you know, the right trails, you know, and not getting as distracted by the rabbit trail, as, you know, my family might say from Kentucky. But anyway, this is good. These are all positive steps in the right direction. And then, as I already mentioned, the tumor subclassifications. It's a lot of animation. I apologize. So again, you know, it's a big effort. You know, we want to understand again. You know, colon cancer is not colon cancer. Is not colon cancer. You know, pancreatic cancer is not, you know, pancreatic cancer. These are different. You know, each tumor is very different. And we want to, you know, understand that at a much deeper level, at a very genetic level, submolecular level. And that's what we're driving for. And then, you know, finally, you know, talking about the understanding response to therapy. You know, obviously this is a goal. You know, we want to understand. You know, you've heard immunotherapists talk about a lot. You know, we want to understand how to rev up the immune system when it's appropriate. You know, without causing more damage, and not, you know, doing that in cases where a patient would not benefit. So again, you know, this is kind of, you know, some of these you know, these overarching categories, you know, some of these big categories that, you know, we are making progress in. It's complex, but, you know, again, you know, we are making strides in the right direction. So I'm from Illumina. You might be wondering what the heck does Illumina do? You know, what are we known for? What are we famous for? So I would say what we're famous for, or best known for, are our sequencers. And you might be saying, well, what's a sequencer? And a sequencer is a big box with a lot of computational hardware, moving parts. But basically what it allows you to do is take a sample of DNA, like a tumor sample or a blood sample, and you mix that with some reagents, and you kind of plug it into the machine, and it spits out a lot of information. You know, so it spits out your, you know, the genetic sequencing that's associated with that tumor or that sample. So Illumina kind of made its name with these big, expensive sequencing machines, you know, and they're very expensive. You know, Foundation Medicine has its share of, you know, the big sequencers, and these really, you know, the really top academic medical centers have these big sequencers, but they're cost prohibitive. They're very expensive. They're expensive to run, but they're very purpose-built. And what we've done over time at Illumina is we've gone from making these giant, cost-prohibitive sequencers to now we have these tiny ones. You know, I mean, this thing is a beast. You know, it's massive. It's even bigger than a refrigerator. And now we have these tiny ones. You know, it's like this little bread box kind of thing that you can sit on a bench top, and it's much cheaper. You know, so the great thing that it allows, you know, A research lab that can't afford the big one is they can afford the small one, and they can do just as good, research in their own smaller, academic centers and contribute, to this conversation, or contribute to the research, which is critical. We want to democratize this so that anybody that can contribute, has the ability to do that. So this is something I'm very proud to be, part of a company that's allowing, this kind of thing to happen. So. And then ultimately, those discoveries, the research that's being done, the new pathways that are being uncovered. We want to take that and we want to translate that and we want to apply it. We want to bring it into the clinic. We're a company, we're not physicians, but we want to create tools and we want to provide, power and empower physicians to make the best decisions possible. With that. So that's ultimately the goal. And obviously we've seen Foundation One, they're applying the technology in a really great way. So again, this is the idea that we kind of hold hands with scientists and clinicians and pharma companies, using technology, cutting edge new technology, to better diagnose, to better understand and give prognosis. And somebody raised the question earlier about sample. Sample can be tough to get, especially in research. Believe it or not, even in conditions where there's a lot of tissue available, it can be very difficult to obtain. And it's precious tissue. And it's difficult, to share tissues, between different academic medical centers or especially cancer centers. And again, going back to this idea that it's very precious. And Sherry, she talked a lot about lung cancer, and I did a lot of work on non-small cell lung cancer, and the biopsies are really, unfortunately, very complex. So you have to open somebody's chest just to get a needle biopsy. I mean, this is a needle core. This is a needle that you're pushing into somebody's chest, into their lung, and you get this incredibly small amount of tissue out, and then that's it. That's all you have. So you have to make the absolute best use of that small amount of tissue that you can. And you want to test as many variants as you possibly can. Again, Foundation, they have hundreds. And unfortunately, there's a challenge in the field right now that a lot of labs just do single tests, single gene tests, and it burns a lot of tissue for that single gene test. And that's why we're so excited and want to proselytize our technology, because you can take very small, these are 10 nanograms, versus some of these bigger quantities. A very small amount of tissue sample and get an incredible amount of information about it. And that's what we're hoping, that's what we're pushing for. Those are the strides that we're making today. And we continue to work toward. And this is just an example. We have a lot of assays, and again, we're proudly partner with Foundation One to work with those guys. But again, turnaround time, and having accurate results. I'm not really going to speak to this slide too much, but the real idea behind this is you want timed results, right? You don't want to have to wait, days or months. Again, as Sherry pointed out, if you do a single, you're doing a single gene at a time. You're testing a gene with a single gene at a time. If you don't run out of tissue first, it's going to take months. Sometimes, unfortunately, to really get to that. What gene is mutated? Is that druggable? Why not have hundreds of genes, in a couple of days? So cost savings, and then we want to maximize the results with minimal amount of tissue. Ultimately, to better patient stratification and treatment by our physicians. And we do, I don't know why these red boxes are there, I apologize. But we do offer, assays again, just very similar to Foundation One. But again, moving from single genes and to hundreds, if not thousands more genes. That's our goal. Is to take blood samples or solid tumor samples or bone marrow samples and be able to, in very small quantities, and be able to run hundreds, if not 1,000 plus genes against that to see where the mutation's at. And usually, again, there's not going to be a single mutation. There's going to be, dozens of mutations. And just to kind of explain the power, you hear these numbers like 1,000 genes or 500 or whatever. We have one product that's actually fairly popular. We call it the TrueSight Tumor 15, and it just assays 15 15 genes, but just having 15 genes, just moving from a single gene to 15 allows you to cover 7 really common types of cancer pretty well. It doesn't cover all the ultra-rare mutations, but it really covers 7 the common mutations very well. So that just gives you the scale of power going from single gene to 15 to 1,000. That kind of scale is just pretty remarkable, and the power and the information that it provides you with. So this is my last slide. Ultimately, again, the goal is always better decisions in the clinic. We want to help, or we want to have patients and clinicians understand their genetic makeup better, understand mutations, understand how they're responsive to a particular drug, stratify those into a particular targeted drug. Hopefully one day getting away from chemotherapies that that target just broadly target rapidly dividing cells. And you can really focus on single molecules, and minimize the side effects. And obviously, putting those folks with the right type of drug, through clinical partners. At Illumina, we partner with a number of pharma companies, Amgen, AstraZeneca, Merck, Sanofi. So these are some of the big drug makers out there. So very proudly work with those guys. I'm on a project right now that works with Amgen. So yeah, I know that's very high level. I know most of you guys are incredibly educated about all of this, but if you have questions, I would absolutely love to answer them. Yes?
So could you please, I'm a little confused, contrast, are you a tech company that has the physical means to do this?
Sorry, yeah.
And how do you differ from Foundation 1?
Right. So I guess you can think of it as we provide tools. So we provide like the sequencers. We also provide assays, that researchers can use. That these are pre-built assays. It can be difficult to build them on your own. And so that's what part of my job is, to develop these cancer assays and market them to understand what the need is, and try to get as many relevant mutations in particular assay as possible.
And then you partner with the medical companies so that they can maybe get a jump ahead to address these specific genomic mutations.
Exactly. That's exactly right. So we partner with Foundation Medicine. We're essentially a tools provider for Foundation.
Okay.
They're very smart folks. They kind of do their own thing.
But they use your.
But they use our boxes and our reagents. But sometimes the pharma companies want something that's a little bit more of a complete solution. Again, Foundation, they do amazing work. And but again, a little bit of a different partnership.
Right.
Sometimes we help pharma companies with, in clinical trials or that sort of thing. To help get them that information.
Sure.
Hopefully that makes sense. I apologize if I wasn't clear earlier, but that's kind of what Illumina does. We provide the tools. We build assays. We work on a lot of partnerships, because these pharma companies, they don't always know what to target. They don't know if in colon cancer or pancreatic cancer or choose a cancer that they don't have great ways to understand what those mutations are without this kind of technology, without these types of assays. So we try to do those partnerships, and if they see for instance a mutation that comes up over and over and over and over again, and there's good evidence that's a driver mutation, then they certainly would want to make a drug.
Medication, sure.
Exactly. Hopefully that was coherent.
No, don't just blame your baby.
Yeah.
Thank you.
Yes, please.
I've got a quick question.
Yes.
Have you ever thought of keeping the data that your machines produce? It seems like there's an opportunity for big data and analytics to take a look at the results from all the different tests that are.
That's brilliant, actually, and I can't say too much about it, but absolutely, because big data, that's what we want. In the end, in today's world, a lot of the institutions, they have their own library of data, and part of the reason they keep it is because it makes them valuable.
Right.
It's like we have this special knowledge of these particular types of cancers. We know how to treat it, and unfortunately, to give that information away takes away some of their competitive advantage. If we could get everybody to share everything, that would be amazing, and I think that would, be a faster way to, to drug treatments. But yeah, that's a great point. I wish we could talk a little bit more about it, but yeah, you're absolutely right. We would, yeah, love to do that.
I have a question for the general, everybody here. How many of you have actually had genomic testing on your tumors? How many? Everybody who's had that, who's actually received targeted treatment for your genomic testing? One?
We select, I think probably similar to you, we selected chemotherapeutic agents that seem to be more effective.
Okay.
And unfortunately, targeted therapies is that term, that classification is still in its infancy. We only have a handful of targeted therapies now, and it's been, they're kind of picking low-hanging fruit so far. But as somebody said, there are 800 compounds that are targeted therapies that are in trials right now. So it should get much better. Unfortunately, it's going to take time and money to get there.
The two that raised their hands, are they on the same drugs, the chemotherapy drugs? Regular, not?
We went for fulfuriate after oxaliplatin because I couldn't tolerate oxaliplatin anymore, but we chose that you're already taking it based on.
So it wasn't like a targeted treatment like it meant, it was actually a general chemotherapy?
No, I used Erbitux before we had the knowledge that wild type versus non-wild type was significant.
And same for me, it wasn't like it was a refined
It wasn't a targeted
treatment, it wasn't a targeted therapy, it was more which hammer to use.
Okay.
But I just also working for Illumina, I can say 10 years ago when these guys were going through this process, the cost of genetic sequencing and learning about the DNA of your tumor was pretty prohibitive, right? We paid out of pocket for quite a few things, and we had, we had the means to do so, but what Illumina is doing is making this accessible to most everyone.
I think that's
right?
great. I think we as a group should be able to pull that information across all of us as a group, to use as a group for treatment options as a group. And then have a doctor sponsor that locally, wherever we live. Personally, I don't know how you all feel, but.
So Chris, that's your action item.
Super easy.
I think we've got the right people to start that conversation in this room today, so I think that can happen.
So when do we get our little kids like Ancestry.com does?
All right, now it feels like every other Sunday at 3 o'clock when you have our team call. Okay, give me a couple weeks, we'll work on that. Before you know, we'll be ready. I love it.
Believe it or not, that it may happen sooner than we
Yeah.
can. So.
Jeremiah, thank you so much.